Hello, I am ZAFIRAH along with my partner Lim Jian Xin, and we are from Group 17. This is a summary of the article Recent Advances of the Machine Learning Methods in Predicting NCRNA-Protein Interaction by Lin et al. So this video will be divided into 5 sections, which are the introduction, data sources, the performance and evaluation, computational methods, results and discussion. Study on RNA has been receiving attention since Andrew Fall and Craig Mallow achieved the Nobel Prize for their RNA interference, gene silencing by double-stranded RNA in 2006. RNA can be classified into coding RNA and non-coding RNA. This article will be focusing on the NCRNA. NCRNA are the RNA that cannot keep a code for proteins but still fulfill their role at RNA level, and most of them have low molecular weight. Some examples of NCRNA are ribosomal RNA, rRNA, transfer RNA, tRNA, microRNA, miRNA, small nuclear RNA, snRNA, and long non-coding RNA, lncRNA. RNA are found to be involved in various functions like participating in the process of protein translation, and they are one of the most important molecules in RNA implementation. They involve in gene expression, chromatin modification, cell differentiation, and epigenetic regulation. Researchers found that recognizing the interactions between NCRNA and proteins will help in understanding the functions of NCRNA. However, the research to find NCRNA functions founding needs time, money, and energy. Therefore, it is more practical to use computational models to assist in the research process. Machine learning is an advanced field that investigates the use of computers to mimic human learning activities. This field searches for the basic laws of data and unknown data that are predicted using the combinations of relevant data and algorithm. Machine learning can be classified by the learning methods that are supervised learning, unsupervised learning, semi-supervised learning, and reinforcement learning. It can also be classified by the algorithms that are decision tree algorithm, supervector machine algorithm, random forest algorithm, and deep learning. This article reviews on few NCRNA proteins interaction models that utilizes machine learning. The specialties of each models and limitation of the machine learning-based methods are also discussed in this article. Let's move on to the data sources. NCRNA protein databases and datasets are set up to further understand the NCRNA protein interactions and to assess the performance of the methods. RPI-488 is the dataset of the non-redundant LNC-RNA protein interactions in reference to the structural complexes that has 488 LNC-RNA protein pairs. RPI-2241 is the dataset almost similar as RPI-488 but with 2,241 LNC-RNA protein pairs. NPI-Inter version 2, on the other hand, is the database of NCRNA protein interactions that are not structural based with 10,412 NCRNA protein pairs. The last one is the RAID version 2, which is the database of biomolecule interaction information that includes a great portion of NCRNA protein interaction. The performance of the models for prediction are assessed commonly by leave-one-out cross-validation (LOOCV) and K-fold cross-validation (KCV). In KCV method, the data will be split into K subsets of the same size randomly. One subset will be the testing set and the others will be the training set in every evaluation. The testing and training subsets will not overlap to avoid bias comparison, and the final result will be the average of the K test results. LOOCV method uses the basic concept of KCV method, by which the K data will be substituted into M samples. One sample is utilized as the testing set and M-1 data will be the training set. The performance of the prediction models will be quantified by the average of M results. LOOCV utilizes most of the samples for model training, and therefore the finalized assessment are more credible. Cross-validation is applied in LOOCV to generate the receiver operator characteristic ROC and the area under ROC curve AUC. The classification model is evaluated using AUC, which is a significant index. The perfect classifier will get 1 as the AUC score, and the AUC for random classifier is 0.5. The first computational model is IP-minor, and it is used to discover the unexploited interaction modes between NCRNA and protein. 3 mere characteristics of protein sequences and 4 mere characteristics of RNA sequences are firstly obtained to generate the 343-dimensional features and 256-dimensional features respectively. Stack autoencoder is used to generate the high-level features from the protein and RNA features, and this is known as SDA. SDA-FT is obtained by fine-tuning the stack autoencoder using stack information of the training data. RF classifier will be used to generate 3 predictors that are RPI sequence RF, SDA RF, and DSTF RF using SDA FT and SDA features. Stack ensembling is used to obtain the output data of the predictors. Output level 0 classifier is the predicted ability score, while output level 1 classifier is the logistic regression. IP-minor has an accuracy of 0.891, sensitivity of 0.939, and precision of 0.945. The scale of NCRNA protein detection and the use of manual labor in organizing different sources features are greatly improved by using the stack autoencoder. The disadvantage of IP-minor is that the high-level features that are automatically captured by deep neural networks are not elaborated clearly in the biological standpoint. LPI PPSN computational model fuses 4 protein-protein similarity networks PPSN to predict the LNC-RNA protein interactions. These PPSNs are obtained by calculating the protein sequence, protein domain, protein functional annotations, and string database. The similarity of any pairs of proteins is calculated by using different similarity metric before generating the protein-protein similarity networks. 4 PPSNs are fused together by using the similarity network fusion SNF algorithm to obtain informational PPSN. Non-LNC-RNA protein interactions and fused PPSN are the base of generating a heterogeneous LNC-RNA protein network. The new LNC-RNA protein interactions on the heterogeneous network are predicted using the HATI-SIM algorithm. The AUC for the fusion of 4 different protein-protein similar network model is 0.9068. The accuracy of LNC-RNA protein interaction is improved as the number of fusion matrices increases, and this will generate a more specific and comprehensive protein similarity network information. The BLP-ISH computational model is used to predict the interaction of LNC-RNA protein by using the HATI-SIM score. LNC-RNA similar network, LNC-RNA protein association network, and protein interaction network are used to generate a heterogeneous network. HATI-SIM metric is used to calculate the similarity score and eventually obtain the HATI-SIM score of every LNC-RNA proteins for each path. The HATI-SIM score is then used to obtain the SVM classifier to predict the interactions between LNC-RNA and protein. The AUC for PLP-ISH is 0.95. And the advantage of this computational model is that the indirect biological network topology about LNC-RNA is included, which is vital in deducing the novel LNC-RNA protein interactions. The next computational model is HPLI ensemble, which is particularly used to study human LNC-RNA protein interactions. HPLI assemble is used to generate HPLI SVM assemble, HPLI RF assemble, and HPLI XGB assemble using an integrated strategy that are based on SVM, RF, and XGB. Three NCRNA feature combinations are obtained by using three different feature extraction methods, which are KMIR, DAC, NPC, PSE, DNC general. This method is repeated to get the protein feature combinations, which only DAC feature extraction method is changed to AC feature extraction method. All these feature combinations are combined before they are used to train another 3 machine learning algorithms, SVM, RF, and GB, to generate 27 submodels. The integration strategy that suits the HPLI ensemble model are selected from the average, and the linear integration strategy based on the performance of the integration strategy. HPLI ensemble is better than the previous model, as HPLI SVM ensemble, HPLI RF ensemble, and HPLI XGB ensemble reach an AUC of 0.95, 0.96, and 0.96 respectively. The advantage of this computational model is that it is successfully decreasing the false positive rate and restricting model overfitting by using ensemble strategy and random pairing strategy. However, the limitation of HPLI ensemble is the lack of known human NCRNA protein data and the enormous computing resources.
Besides that, another type of deep machine learning is RPI-SUN. RPI-SUN model utilizes SUN or stacking autoencoder networks to extract more specific features from protein sequence and non-coding RNA. These features are then inputted into the random forest model to forecast the possible interactions between non-coding RNA and particular protein. Based on the flowchart shown in Figure 5, there are two ways on how the RNA-protein pairs are processed. First, the position-specific scoring matrix, also known as PSSM, and Kramers sparse matrices are utilized in order to isolate the useful properties of corresponding RNA and protein. Following that, each of the RNA sequences that were inputted is converted into a Kramers sparse matrix that is able to save practically of all the information on the order and the composition of amino acid. The method of singular value decomposition, or SVD, is then employed to derive vectors from each matrix. On the other hand, PSSM incorporates evolutionary data with position-specific recursive BLAST to transform the protein sequence to PSSM. From PSSM, the program extracts pseudogenic moment PZM characteristics, as it can acquire metric characteristics with minimal redundancy. After that, the model leverages SAN to intuitively discover concealed at-once characteristics from the features. The sparse autoencoder that makes up the SAN is a multi-layer neural network where the output of one layer is used as the input layer for the layer that comes after it. Both high-level features of RNA and protein are generated. In the final step, the RF classifier receives these representative properties as the input in order to make the prediction regarding the non-coding RNA-protein interactions. The accuracy of the prediction of this model can be enhanced by applying layer assembly, which is applied in the following use. Another sort of machine learning is LPGNMF. LPGNMF is used to find unrecognized interactions between long non-coding RNA and protein. Compared to the other machine learning, it uses matrix factorization method to reduce the matrix into each constituent part to anticipate the association between long non-coding RNA and protein. LPGNMF model comprises 3 steps to run. In the first step, long non-coding RNA and protein resemblance is evaluated according to long non-coding RNA expression and gene ontology data. Then, Pearson correlation coefficient (PCC) is used to assess the similarity of long non-coding RNA, whereas the degree of similarity between every 2 different proteins is evaluated by GI². Next, it uses the nearest neighbor algorithm to generate the long non-coding RNA bipartite graph and long non-coding RNA similarity matrix yields the weight matrix. It is the same applied to the protein sparse similarity matrices. After that, both long non-coding RNA and protein sparse similarity matrices are integrated, and the relationship is estimated by applying non-negative matrix factorization and Laplacian regularization algorithm. Finally, the matrices employ an iterative approach until conversion is achieved, and the long non-coding RNA and protein interaction score matrix is calculated. In this model, each long non-coding RNA is used to analyze protein by using graph-regularized non-negative matrix factorization. BGFE is another form of deep machine learning to predict the relationship between non-coding RNA and protein that employs the stack autoencoder network as well as the random forest classifier. For non-coding RNA sequences, K-MER matrix is used to model the sequence and SVD is used to generate them into corresponding vector characteristics. While for protein sequence, PSSM is used to mine the evolution information of every protein sequence as additional data. After that, both processed non-coding RNA and protein are included into the layered autoencoder, in which the output layer of each layer is connected to the input of the subsequent layer. Following that, Kuwait-Leiber divergence score is employed to compare the predicted and actual scores. Ultimately, the calculated information with the labels are merged into the random forest classifier to systematize if there is interaction between non-coding RNA and protein. The advantage of this model is that the stack autoencoder can automatically learn the mask association between sequence components. Therefore, it can improve the overall performance of the model. Moving on to the next deep machine learning model, RBISE. This model combines stacking ensemble computationally operation, gradient boosting decision tree, support vector machine, and extremely randomized tree to forecast the protein and non-coding RNA sequence correlations. In the first phase, RBISE leverages non-coding RNA sequences by adopting the K-MER sparse matrix, and they are subsequently processed by SVD to decrease the input vector dimension. However, Lejeune movements are used to derive the data into the vector form from protein sequences position weight matrix. After that, the machine learning algorithms such as XGBoost, SVM, and Extra3 are supervised with the attributes mentioned. Each feature plays different roles to improve the performance of the model. XGBoost establishes regulation in the cost function, which is beneficial to minimize the variation of the model, overfitting and limiting the degree of complexity. In other words, the cost function evaluates how inappropriate the input-output relationship the model finds after it is trained depends on the ability of predicting. For SVM, support vector machine is a linear and sparse binary model that can simplify the problems of classification and regression. For solving learning samples, this method produces a line or construct a hyperplane, and its decision boundary is the greatest margin hyperplane. Besides, the radial basis function RBF kernel is also incorporated into this machine learning, where it helps in decreasing the intricacy of arithmetic algorithms. Extra3 classifier is an example learning method that builds decision trees, and each extra tree is built from the training sample. Extra3 constructs completely randomized tree by randomizing features and cut point decision while splitting nodes. It can typically decrease the model's variance but can resulting in bias. Lastly, the stacking example is utilized to include these machine learning algorithms to form RBISE model. The prediction performance of this model is enhanced by combining all the machine learning through the stack example, as each distinct benefit is provided by the machine learning. For instance, XGBoost has a better prediction accuracy features, whereas SVM has better stability prediction features. The final computational model mentioned in the article is LBI-MMHN. It is a newly developed model that utilizes microRNA to act as mediators to forecast the association between long non-coding RNA and protein. Unlike other models, this model is different as it does not depend on the well-defined long non-coding RNA and protein interactions. It considers the amount of distinct long non-coding RNA, amount of common microRNA, and amount of different proteins to assess the interactions. This model is then evaluated by determining the resemblance of long non-coding RNA or proteins in three distinct methods, namely network similarity, sequence similarity, and statistical feature similarity, which multiple bipartite graphs are used that represent the network interaction between long non-coding RNA with microRNA and network interactions between microRNA with protein. The same operation also applies to protein similarity networks. The network similarity is assessed by using the algorithm defined by Smith-Waterman. Additionally, the statistical feature similarity is operated where the composition of pseudo amino acid is used to describe protein sequences, whereas the composition of pseudo amino acid is used to represent the protein sequences. The normalized similarity matrix of long non-coding RNA and protein are formed respectively after integration of the outcomes generated from distinct methods. After that, a model of a heterogeneous network is created by condensing the bipartite graphs of network interaction between long non-coding RNA with microRNA and network interaction between microRNA with protein. Finally, the prediction score matrix of long non-coding RNA and protein interactions can be evaluated by integrating the similarity matrix of protein, long non-coding RNA and the heterogeneous network. This model is valuable for researchers to discover the unknown relationship between long non-coding RNA and protein that provides the knowledge regarding its particular function. Moving to the result, the performance of each machine learning models, which are IP minor, LPI PPSN, PLPI-HS, HLPI assemble, RPI send, LPGNMF, BGFE, RPI-SE, and LPI-MMHN are quantified by cross-validating MP-Inter v2.0, rate v2.0, RPI-1488, and RPI-2241. The performance value indicates the model exhibits great precision and capacity in protein and non-coding RNA association prediction. Based on the result tabulated above, IP minor scores 0.995 value in AUC when cross-validated with MP-Inter v2.0 dataset, and it is evaluated by 5-fold cross-validation. It is better than the other methods of LPI PPSN and HLPI assemble. Besides, IP minor scores 0.914 in terms of AUC, which is validated with the RPI-1488 database. It is not as good as RPI send, which scores 0.920 AUC. There are three models which are validated by the RPI-2214 dataset. RPI-SEN has the best performance as compared to IP minor and BGFE, in which the AUC value for each particular model is 0.962, 0.906, and 0.947 respectively after evaluating by 5 cross-validation. For LPI-MMHN, the value of AUC is 0.821, which is validated with rate v2.0 dataset and is evaluated by 5-fold cross-validation, and this indicates that this strategy complements to others can improve the performance strength. The limitation of this research is that a less amount of training samples and the negative control data set is used. IP minor overfits, resulting in poor performance on the MP-Inter v2.0 database, and AUC cannot adequately quantify the model's good performance. Hence, the further totally independent test sets should be created to effectively compare the models. Proceeding to the discussion, the use of computational models to predict the function of non-coding RNA is increasingly favored by researchers, as it can discover the unknown non-coding RNA-protein interactions. However, there are some limitations in the models. The limitations include that the information regarding the relationship between non-coding RNA and protein is limited, and therefore only a small data set can be utilized to train the computed models. Besides, the machine algorithms provide advanced features that cannot be described and explained biologically. For certain models, acquiring negative examples, selecting appropriate parameters, and combining classifiers present formidable obstacles. There are some recommendations to improve the performance of models. One of the recommendations is that a detailed database of non-coding RNA protein associations must be established. Besides, vast quantities of non-coding RNA protein association data can help in the development of more accurate prediction models and increase their reliability. Not only that, deep learning methods that have the characteristic to automatically derive advanced features from raw data to facilitate the prediction of interactions between protein and non-coding RNA. Since the models discussed utilize the IOC curve and AUC to assess the performance, which may not completely reflect their advantages and disadvantages. Consequently, the use of additional assessment indicators such as the Gini coefficient, leaf curve, and gain curve may improve the prediction performance of a model. Besides, building a better network architecture to discover the advanced functions of useful biological knowledge is the recommendation to improve the performance. As a conclusion, machine learning-based computational models that predict non-coding RNA protein interactions are detailed with the computation phases. Besides, we also discuss the pros and cons of each computational model and several complications of the machine learning model. Yeah, that's all from us. Thank you for listening.